Let's begin. Remember to put in a comment about the quiz. I've got two options. One is we can take it from 8 p.m. Tuesday night till 8 Wednesday. It means you must finish by 8 p.m. on Wednesday. The other alternative is we can use another day. There's one student who's requesting that we change having the quiz on Wednesday. And so I'm having a discussion about it. And you can see it, I think it's under week 2 lecture, maybe it's week 3 lecture, something like that under the main page. Can you guys hear me now?
Yes.
Yes.
Great. All right, let's begin. Let's see. Share my screen. Turn on the sound. Hopefully you're seeing my screen now. Let's see, where do I want here?
Yes, we can see your screen.
All right, so we're supposed to be working on chapter 3 today, and actually we're supposed to finish it. I don't think we'll get that far along, but we'll see.
Oh, wait, before we
there's no
start,
lab today. Sorry, what?
could you go over the last question of quiz 1B, like the difference between the size of a virus and the size of a protein?
Quiz 1B. I don't remember that question, but it's the size of a virus and the size of a protein.
Right, like the difference between the two.
All right, well, a virus would be larger because it's more than just protein. A virus is at the very minimum. It has nucleic acid as its genome, and then it's surrounded by a protein coat, which is usually more than one protein, but on some viruses, it's the same protein and just repeating. But it's more than one copy of that protein. And then a protein would be smaller because it's one protein. Any questions about that?
That's clear. So a virus is multiple proteins around some kind of DNA
Around
thing,
the
right?
nucleic acid,
Yeah,
yes.
the nucleic acid.
Yeah.
Okay,
Some
that makes
viruses
sense.
also have a lipid membrane, or not a lipid membrane, lipid envelope. And we'll talk about that when we talk about viruses later in the term. All right, let's go to chapter 3. So we're going to be talking about observing microorganisms. This slide is showing you the major goals of this topic and a rough outline. Know the terms, what's resolution, refraction, stains, basic and acidic dyes, and you can use stain or dye interchangeably. Smear preparation, mordant, up to the glycan. Understand the sizes of organisms and the properties of microscopes. Understand the major differences between light microscopes and electron microscopes. Understand why staining improves resolution. And know that there are 3 types of stains: simple stains, differential stains, and special stains. You should lastly understand the Gram stain, its procedure, and the basis of its differential staining. Now, I'm not going to talk about this much in the lecture, but in the web page, I have links to help you review the metric system, and they're online quizzes which don't have anything to do with Canvas. They're just a link on the web, and generally to operate them, because it doesn't run in Canvas, you click the link and then it gives you another page to click the link or something like that, and that takes you outside of Canvas. But we're going to be learning about the metric system. It is a unit of measurement, and it's a very good unit of measurement in the sense that everything is based on a multiple of 10. For measurement or length, we have the meter, and a meter is made up of 1,000 millimeters. 1 millimeter is made up of 1,000 micrometers, or we could say 10 to the 6 micrometers equal 1 meter. 1 micron is 1 times 10 to the negative 6 meters, or 1/1000th of a millimeter. And then there's a nanometer. This one is one you probably should know because this is the size we see under the electron microscope. I should mention a micrometer is the size you see under a high power, oops, I didn't mean to do that, high power of the light microscope, and then high power with the electron microscope is 1 nanometer. You can go below 1 nanometer, but this is the general size of things we see under the electron microscope. And you don't need to know all the conversions, just if you know, let me blow this up. You should probably know all of the units on the left here. So a kilometer is 1,000 meters. Kilo means 1,000. Then you should know a meter. There are units in between, like a decameter and a hectometer. You don't need to know those, but 10 meters equal a decameter, 10 decameters equal a hectometer, 10 hectometers equal a kilometer. And like I said, the metric unit is based on 10, but you should know that 1,000 meters equals 1 kilometer, meaning you should know these. The meter is the base unit in metrics for measuring length. You don't really need to know this one, a decimeter, 1/10 of a meter. A centimeter, you should know that one, 1/100th of a meter. A millimeter, 1/1000th of a meter. A micrometer, 1/1,000,000th of a meter, or 1 times 10 to the negative 6 meters. And then a nanometer, 1/100000th of a meter, or 1 times 10 to the negative 9th meters. A picometer, you don't really need to know. Any questions about any of that? All right, so this is for length. Metric uses a gram for weight or mass. And then you use the same units, like a kilogram, a centigram. And then for volume, you use a liter. A liter is about the size of a quart. I think it's a little smaller than a quart, but about the size of a quart. And then you can have kiloliters and centiliters. Any question about any of that? Why do you need to know the metric system? Because the metric system is used in science and medicine. And you see all these countries in the world colored? These are the only countries in the world that do not use the metric system. So you should learn the metric system because almost everybody in the world uses it. And there's only one major exception, the United States, that does not use it. And if you don't know, the United States is slowly converting to using the metric system. And that conversion started when, I don't remember, it was Thomas Jefferson or one of the presidents, one of the early United States presidents. But the United States passed a bill that we will switch to the metric system. And we're slowly converting. You can see on your cars that they will tell you, at least a modern car, how fast the car is moving in miles per hour, but it'll also tell you how fast it's moving in kilometers per hour. And some road signs tell you both too. Any questions about the metric system? All right, you should know that a eukaryotic cell is larger than a prokaryotic cell, and the exact range of a cell can vary depending on who the author is, but we're just going to go with this. But the important thing to know is the eukaryotic cell is on average about 10 times the size of a prokaryotic cell. Any question about that? About an average small eukaryotic cell is a red blood cell. The largest eukaryotic cell that you guys have seen is the yolk of a chicken egg. Obviously, that's much larger than 500 micrometers. And if any of you have seen the yolk of a turkey or an ostrich egg, that would be even larger. Currently on Earth, the largest cell is the yolk of the ostrich egg. But none of you have seen that. Well, probably none of you have seen that. I've never even seen that on television. A prokaryotic cell is around most of them, 1 to 2 micrometers, but there are some that are considerably larger, up to 10 micrometers. And this is just a range. They can get smaller than this. The smallest prokaryotic cell is about 0.4 micrometers. And a virus is about 10 times smaller than a prokaryotic cell, with their diameter ranging from 0.02 micrometers to 0.44 micrometers. And you'll notice that the largest virus is about the same size as the smallest prokaryotic cell. You can also, measure viruses by their length. And as you can see, the length of the smallest virus is about the same size as the diameter, but the length can be quite larger. Caitlin, do you have a question?
Yeah, I do. Since that wasn't covered in the lecture on Tuesday, and we had it on the quiz, I think that's one of the ones that I got wrong for the quiz.
Let me take a look at that. The problem is that, let me go to the next slide here. I did show you this. So if the question was just the size of viruses and the size of bacteria, you should know that from this slide that I've showed you previously. However, if I asked you the specific size, which I'm sure I did not, you would not know the specific size.
No, it was the size ranges of certain stuff.
All right, let me go over that. Can you email me a reminder?
Yeah, I
I
can.
will look that over. Okay,
Thank you.
all right. All right, so you should know the different main parts of the microscope. In the microbiology, we work with the compound light microscope. There is a simple microscope, and did I discuss that earlier? Yes, no, I didn't. All right, so we'll talk about the simple microscope later. The compound microscope has 2 or more lenses that magnify. So it has the ocular lens or the eyepiece, and it has the objective lens, and both of them magnify. The total magnification is the magnification of the objective lens, the objective lens times the ocular lens. So you just take one times the other. The typical lab microscope has 4 objective lens, the 4x, the 10x, the 40x, and the 100x objective lens. Now, in our lab, all of the ocular lenses are 10x. And so the total magnification would then be 10x times whatever objective lens you're using. So if we're using the 10x objective, the total magnification would be 10 times 10 or 100x. Any question about any of that?
I have a question. I was reading on the internet about like how to find fields of view, and it says there's usually some kind of division symbol on the ocular lenses, like divide by 10 or divide by 18 to find field of view. I was wondering what we were supposed to use.
We gave you a table giving you the measurement of the field of view. There is a formula, which was, I think, underneath that table in the lab that I gave you, that you can use to get the field of view. Okay,
So I think on the last lab for the oil immersion, I put 400 and got it wrong. So is it actually 40 then?
what are
Yeah, I
you
put
talking about?
400
The
too.
size of the field of view?
Yeah, it was like it was like the last question. And
The last question was determining how many cells go across the field of view.
oh, sorry, then it was 4K is what it
4K,
was.
okay. The field of view for the oil immersion lens, that's the 100x objective, is a little bit less than 0.2. Is that micrometers? Sorry, I'm losing my train of thought. I think it's 0.2 millimeters. So something like 0.18 millimeters. For the 40x lens, the field of view is something like 0.4, between 0.4 and 0.5 millimeters.
Okay, thank you.
All right. And don't quote me on the exact numbers because I don't remember exactly. I'd have to look it up. I remember the oil immersion lens, but the 40x, I don't remember exactly what it was.
Okay, I
All
pulled it
right.
up and it looks like we divide by 18
For what?
for the field of view thing. Like you take the magnification and divide by 18 to find the field of view, right?
I don't know that. I'd have to look that up and take that. You'd have to start with the correct unit and then divide by 18 because obviously, let's see, for the 100x lens, so you're taking 100 and then divide by 18. No, that's not right. All right. So know these parts of the microscope, the ocular lens. You don't really need to know the body tube, but the arm of the microscope, you should know. The objective lenses, the stage, the clip holder for the stage, the condenser, which is with the iris diaphragm. The iris diaphragm, let me blow that up a little bit. On this microscope, the iris diaphragm is here and the condenser is there. I think on our microscopes, which you won't actually see in the lab, the condenser is below the iris diaphragm, which is above. The diaphragm is a shutter and you can open and close it to allow a different size of the cone of light coming through to the slide. The illuminator is also called the lamp. The coarse focusing knob, the fine focusing knob, and then the light switch. All right. Now you should know that a compound microscope has two lenses to magnify. And this is the lens that Van Leeuwenhoek made. It's actually one of his. And you can see this is a simple microscope. because it only has one magnification lens. And that's probably why his microscope was the best. Not only was he the best lens grinder of his day, but he only used one lens. And so it was better at focusing because the microscope and the lenses weren't perfect. And so they started to blur things a little bit. And if you only use one lens, then you have less blurring. If you have a simple microscope, what it really is a super duper magnifying glass. But the point is, a simple microscope uses only one lens to magnify. Resolution is the ability of the lens to distinguish 2 points as being 2 independent points. The best microscopes have a resolving power of about 0.2 micrometers. Anything closer together than 0.2 micrometers, you cannot distinguish it as 2 points. And the resolution limit of the best microscope depends on the resolution limit of white light. Everyone get that? And you cannot magnify anything closer together than 2 micrometers or magnification beyond that, because white light won't allow you to see it. And I don't know if that's the best way to word it, but it'll do. The best compound light microscope that we can build then, the total magnification you can get is about 2,000 times, or magnifying 2,000 times. Anything above that doesn't do any good because we're limited by the ability of light to magnify more than 2,000 times. Because if you do more than 2,000 times, you're getting at a distance less than 0.2 micrometers. And light won't allow you to see that. A little bit of better description. You can see with this picture that we can distinguish that these 2 points are 2 different objects. But these objects we cannot distinguish as being separate objects. And that's because they're within 0.2 micrometers. 200 nanometers is 0.2 micrometers. So these we can distinguish, and that's what we'd see under the light microscope when looking at this. We can distinguish that one too, because that's beyond 200 nanometers, but we cannot distinguish these 2 or those 2 from each other. And actually all of these are blurred together. Any question about any of that? Now, the best microscopes we have magnify 2,000 times. That's using the 100x objective oil immersion lens, meaning you're using oil, and a 20x ocular lens. And the best microscopes in our microbiology lab only magnify 1,000 times, meaning we do not have the best microscopes. And all of our ocular lenses are 10x. Any question about any of that? All right, you should know that light bends whenever it passes from one medium to another medium, assuming the different mediums have a different refractive index. And that's why it looks like the pencil is bending here, because the air and water have a different refractive index. And so it looks like the pencil bends. Any question about that? And why we use oil on the 100x oil immersion lens is to prevent the refraction of light, because light, excuse me, not light, oil has the same refractive index as glass. Refraction is extremely important for life, because when we see an object in the water, it looks to us like the object is in this position, but in reality, the object is in this position. And so when a bird is looking at a fish and sees it in the water here, the bird wants to catch the fish. It has to know that the fish is actually down here. Any question about any of that? And the reason why the fish looks like it's here when it's rarely there is simply the refraction of light at the interface where the water ends and the air begins. When we're looking at a specimen under the microscope, light coming from the lamp will go through the glass slide, and some of that light will bend, leaving the glass and entering the air. And some of that light will be bent so that the objective lens cannot collect the light. Let's go to the next slide. So here we have the light coming through the slide. Some of the light is bent. Some of the light, you'll notice, doesn't ever get out of the slide, or at least not this side, doesn't get out this side because it's bent this way. But some of it is bent, so it's bent enough so it doesn't collect by the lens. And then, of course, some of the light, although it's refracted, it isn't bent enough and the lens can collect it. Now, why we use oil is because when we have oil here between the lens and the slide, there is no refraction of light. And so this light ray isn't bent, and so it can be collected by the lens. Because this lens with oil collects more light, that improves the resolution of the microscope. The resolution is your ability to see things, how good you can see things under the microscope. And because we're collecting more light, we have better resolution with oil than we have without oil. Any question about that? And that's why we use oil with the oil immersion lens, so that we collect more light, and then we see better resolution. If you don't use oil with the oil immersion lens, you're not going to be able to see anything better than with the 40x lens, and it's much easier to use the 40x lens. All right, any question about oil and resolution? Oops. All right, so bright field illumination is how we normally see things with a light microscope. And it's called bright field light microscope because the object looks bright and the background looks bright. And what you have is a lamp or a light, and the light rays come from the lamp, go through the specimen. The condenser just focuses the light on the specimen. And then the light comes from the specimen to the objective lens, to the ocular lens, and to your eye. So normally in the microscope lab we have, and normally a light microscope is bright field light microscope. A dark field light microscope has a disc in between the lamp and the specimen, so that any light rays directly coming from the lamp do not hit the specimen. all of the light that hits the specimen is indirect, and the lamp ray would normally not hit the specimen, but because of the refraction of light, some of it is refracted back to the specimen, and that is the light we see coming out of the specimen. And so the specimen looks fairly light, but the background is dark because no light is coming directly from the lamp. The opaque disc is stopping it, and that's why the background is dark. And that is called dark field illumination. You only need to know the concept of dark field illumination. You should know the concept of bright field illumination because that's what we use. But you can see the specimen in both cases. Generally, most people consider that this specimen is easier to view than this specimen. Any question about any of that? All right, the electron microscope shown here does not use light, it uses electrons. A beam of electron is focused through the object and collected by the microscope, and then you can see it. And the reason why, I went over that slide. The reason why we can magnify greater with electrons or beams of electrons than with beams of light is because the wavelength of an electron is much smaller than the wavelength of light. And so we can magnify much greater with an electron microscope than you can with a light microscope. All right, any question about that? The electron microscope allows you examination of samples, like viruses, internal cell structures that are too small to see with a light microscope. There are actually several types of electron microscopes. You do need to know about two of them: transmission electron microscope and scanning electron microscope. These are the two main types of electron microscopes. And you'll notice that here is transmission electron microscope and scanning electron microscope, that the objects look different. What do you notice about this object compared to that object?
You don't see what's inside of the one on the left. I mean, the one on the right.
The one on the right, that's correct. With the scanning electron microscope, you're only seeing the surface of the object, but the object is in 3 dimensions. This is scanning electron microscope. This is transmission electron microscope. With transmission electron microscope, you're only seeing the object in 2 dimensions. And we're seeing a plane of this cell, and we're only seeing the cell in 2 dimensions. But with transmission electron microscope, you can focus inside the cell. Okay, any questions? So transmission electron microscope lets you look inside the cell, but it's a 2D image. Scanning electron microscope allows you to see the surface, but it's a 3D image. Scanning electron microscope, where you can see the object in 3 dimensions. Transmission electron microscope, where you can only see the object in 2 dimensions. And this, I think, is the membranes of the chloroplast. There are other types of electron microscope. We're not going to examine you on them, so you don't need to know them, but there is the scanning tunneling microscope. It gives you a three-dimensional image. It uses a metal probe, and it allows you to see 1/100th of an atom. So you can magnify down to very small size. And then there's the atomic force microscope, also not examining, another type of electron microscope, the fourth type. It uses a metal and diamond probe inserted into the specimen, allows you to produce 3D images. And that's what the toxin of Clostridium perfringens actually looks like. All right, why we use a microscope, of course, is to make larger objects which are too small to see with the human eye. And we usually stain the objects to make them more visible. Here you see on the left objects which are unstained, an object on the right which are stained. Both of them are cells, and which cells are easier to view. Come on, cells on the right. Why does staining make the object easier to view?
It
So
helps
they aren't-
illuminate it.
Yeah, it helps illuminate it. What we're doing when we're staining the cells is all we're doing is increasing the contrast between the background of the slide, which is the glass, and the cell. So if that's the same cell, it may not be. We're increasing the contrast between the cell and the background, which is the glass. That's all staining does, increase the contrast. But it makes the cell much easier to view. For some reason, taking 2 slides at a time. So most microbes are colorless. They look pretty close to the same color as glass. You have little contrast with the slide or the medium. To see them, you really do need to stain them. I keep going by 2 instead of 1. Staining, coloring the specimen to increase the background. Before you stain the cells, you generally have to make a bacterial smear. A smear is a thin preparation of what you're viewing under the microscope. And you do want this bacterial smear to be very thin, ideally 1 layer of cells thin, because if you have multiple layers, it won't stain as easily if you have just a thin layer of cells. After you make the bacterial smear, you then let it air dry, and then you heat fix it. And both the air drying and the heat fixing is to attach the cells to the glass. If you don't air dry and heat fix, then in the water washing of the stain, you may wash away all of the cells. Any question about any of that? So usually when we make a bacterial smear, we air dry and then heat fix. Let me tell you just a little bit about heat fixing. You take the slide, pretend this big object is a slide, and you just pass it over a heat, like a Bunsen burner, or with the Bacto incinerator. We'll pretend this is the Bacto incinerator. You touch it to the Bacto incinerator and then just pass it over the Bacto incinerator. And usually you do it 3 times. And that's how you heat fix. You do not want to get the slide hot. You want the slide to be cool enough that you can touch it with your hands and you don't burn your hands. Of course, ideally, you're not wearing gloves when you're doing this, so you can more easily detect the temperature of the slide. But in our lab, you have to wear gloves. And do recall that the gloves decrease your ability to detect the heat of the slide. So once the glass starts getting hot, you're heating it too much. And if you overheat it, you can melt the cells and deform them. Sorry, I was starting to lose my headpiece. So. All right, a stain, also called a dye, is simply a salt consisting of a positive or negative ion which is colored. Remember, a salt is just a substance that breaks into two parts, a positive and a negative part, in water or some other solution. And one of these ions has to have a color to be considered a stain. If the colored portion of the stain or dye is the positive part, then it's the positive stain, and meaning the chromophore is a cation. We call that a basic dye. The colored part of the dye is the positive part. If, on the other hand, the colored portion of the dye is in the negative portion, or the anion, then we call it an acetic dye. Any question about that? So that's
I
the
was
only
just wondering
difference.
why it's like that. Isn't it like usually basic solutions are OH minus and acidic solutions are like H plus?
That's an acidic acidic compound. We're talking about an acidic dye. Okay, and so this may not be releasing OH, and this may not be releasing H plus, although that's positive because it's possible it's releasing H plus because the dye is in the anion, and the positive part, we don't even know what it is, but it could be H plus. Okay. And then this one here, the dye is in the cation, so it could be releasing OH minus, but it may not be. All right. So for this, you don't even need to worry about that. All right. Bacteria have a slightly negative charge. Therefore, a basic dye is better at staining the cell directly. And most, but not all, microbiology stains are basic dyes: crystal violet, methylene blue, malachite green, safranin. And these, I think, are almost all of the stains we've talked about. The exception might be India ink, which is not a basic dye. And the positive basic dye directly sticks to the cell because the cell has a negative charge. Now, the acidic dye has a negative charge and the cell is a negative charge. So that means the acidic dye will not directly stain the cell. They will be repulsed from each other, but an acidic dye will stain the background of the cell instead. Because it's staining the background instead of the cell, we call that a negative stain. And that just comes from old Polaroid film. The negative would be the reverse of what you see in a picture. Let me just show you a negative staining colors the background, but you still can see the cells quite nicely. because they have high contrast with the background of the slide. The basic staining, on the other hand, stains the cells, and you can nicely see them because they have high contrast with the background of the slide. Any question about that? All right. In microbiology, we say there are three main types of stains: a simple stain, a differential stain, and special stains. A simple stain is just an aqueous or alcohol solution of a single one dye. And you can have a single negative dye or acidic dye. This is something I should change. I got it from the publisher who did this. And just whenever you're using one dye, it's a simple stain. The primary purpose of a simple stain is just to increase the contrast between the background of the slide, which is usually the glass, and the specimen. The stain is applied to a fixed smear for a certain length of time. It then may be washed off, dried, and then examined. A simple stain may use a mordant. A mordant is just a substance that intensifies the stain. So the mordant may not directly color the specimen. It is not a stain. It simply increases the affinity between the stain or the specimen, or it binds with the stain, increasing the affinity, meaning increasing the intensity of the stain. It may coat the specimen to enhance it, therefore making it easier to see by increasing it. An example is the flagella stain, which you use a mordant, and the mordant puts layer upon layer upon layer of the primary stain, meaning the stain, the simple stain. And then that layer upon layer allows you to see the flagella. Any question about any of that? A mordant may be used in a differential stain, and actually we've talked about one. Iodine is a mordant in the Gram stain. A differential stain reacts differently than a simple stain, and cells stain differently. So a differential stain reacts differently with different kinds of microbes. And that allows you to distinguish between two different types of microbes. A differential stain usually uses two dyes, but that is not required. The most commonly used differential stain is the Gram stain. And then the second most commonly used differential stain is the acid-fast stain. And these stains stain the cells differently depending on what type of cells they are. The Gram stain, you don't need to know the year it was developed, 1884, by Hans Christian Gram. And it's a very useful stain in bacteriology because you can classify almost all bacteria into two large groups. The Gram-positive cells, which stain purple, and the Gram-negative cells, which stain pinkish. Let's talk about the Gram stain in greater detail. You first make a bacterial smear on a glass slide. You let it air dry and then heat fix it. You then flood the slide with crystal violet. And this is the primary stain. I'll talk more about that. And you do that for 1 minute. You then dump off the stain, crystal violet, and then wash with cold water gently, so you don't wash off the cells. You then flood with iodine, which is a mordant, for 1 minute. And you dump that off and wash the slide with cold water. You then flood the slide with acetone and ethanol, which is a decolorizing agent, for about 5 seconds. The best thing to do is to watch it while you're adding the decolorizing agent. And when the color stops running, stop applying the decolorizing agent. You then need to wash immediately with cold water. You have to wash immediately because the acid and ethanol of the decolorizing agent can continue to work. So you wash immediately. Whereas all the other steps, it's not important whether you wash immediately or not. But here it is. Then you flood the slide with safranin, that's the counter stain, for 1 to 2 minutes. I'd say 1 minute, and then everything is 1 minute except for the decolorizing. And then after 1 minute, you wash, you dump off the stain, and then wash with cold water. You then blot it dry and can examine it under the microscope.
I have a question.
Uh-huh.
So during the procedure, how does it like not wash away the bacteria?
It's because you air dried the bacteria on the slide and then heat fixed the bacteria to the slide. So
Oh,
the cells do not wash off when they've been air dried and heat fixed.
okay.
Okay. All right. So when you apply the crystal violet, you'll note that all the cells stain bluish purple. And then when you apply the Graham's iodine, which is an mordant, the cells will intensify their color, meaning they might be a little darker, and they also might be a little bit more purple as opposed to a blue purple. But other than that, the iodine doesn't change too much what you see with the crystal violet. It just intensifies the dye. We'll talk more about that. You then flood the slide with the alcohol wash, the decolorization step, and that will wash away the crystal violet from the gram-negative cells, so they will look clear. The gram-positive cells retain the purple color, and then we flood the slide with safranin to give the transparent cells, which are clear, a reddish or pinkish color. So the differential step in the Gram stain is the decolorization step. At this point, we can see differences between the gram-negative cells and the gram-positive cells. Any question about any of that? All right. So when you're making the bacterial smear, make sure that the cells are thin. And if it's for the Gram stain, you should also make sure the cells are newly grown. If they're over 2 days old, the Gram stain may not work. If it's for another stain, you don't have to have that requirement that the cells are 2 days old or younger. You then allow the slide to air dry, and then you heat fix the slide. You flood the slide with crystal violet, which is the primary stain. It's called the primary stain because it's the first stain you use, and it colors all the cells. What's happening is the crystal violet is attaching to the peptidoglycan in the bacterial cell wall. You then dump the crystal violet off and wash the slide with cold water gently. The gram-positive cells and the gram-negative cells both stain purple after the application of crystal violet. We then add iodine to the slide. Iodine in this case is a mordant. It complexes with the crystal violet, increasing the affinity of the crystal violet for the specimen. So because the iodine is binding to the crystal violet, we now have a larger molecule, we'll call it that, which is giving color to the specimen. So that's what the iodine is used. Now, iodine, if you don't know, could be used as a dye, like when we're staining plant cells for starch. Iodine can be used as a stain in that case, but in this case, we're just using it as a mordant. You apply the iodine for 60 seconds. You then dump the iodine off and then wash the slide with cold water. After you've applied the iodine, the gram-positive cells are purple and the gram-negative cells are purple. And they may be more darkly staining than crystal violet alone. This is crystal violet and iodine complex together now. All right, after you wash the iodine off, you then flood the slide with the decolorizing agent. And there is more than one decolorizing agent. Usually we talk about acid ethanol, but you could have, I say acid ethanol, but you could have acetone ethanol. In the lab last time, they only used ethanol. So like I said, there's at least 3 different decolorizing agents. We're going to talk about either acetone ethanol or acid ethanol because you apply them the same. The decolorizing agent, regardless of what it is, washes the crystal violet out of the gram-negative cells, out of their cell walls, but not out of the gram-positive cell walls, as long as you don't overwash. So it's considered the differential step in the gram stain because you can see differences after this point. I don't know if you guys can see this. I'll try and put it in front of me and then maybe you can see it. The best thing to do is when you're applying the acetone ethanol decolorizing agent or the acid ethanol decolorizing agent is add it to the slide. And then when the color stops running from the cells on the slide, stop applying the decolorizing agent. Any question about that? You will apply the decolorizing agent for around 5 seconds. And if you apply the decolorizing agent for too long, you will wash the color, meaning the grams iodine and crystal violet, out of the gram-positive cells. So you do not want to colorize too long. If you decolorize too short, you may not wash the dye out of the gram-negative cells. So the decolorizing treatment is the tricky step. You have to decolorize for the right amount of time. Too long, you over decolorize, and too short, you under decolorize. You then need to wash immediately with water because the decolorizing agent on the cells will continue to act, meaning even if you tip the slide to get rid of most of the decolorizing agent, there will be some still in the cells. Any question about that step? All right. After the application of the decolorizing agent, the color of the gram-positive cells remains purple. The color of the gram-negative cells is now colorless. We now stain the slide with safranin. Safranin is a pink dye. It does stain all the bacteria. It is also called the counter stain because it has a different color to the primary stain, meaning crystal violet is bluish or purple, safranin is pink. You apply the safranin for 1 to 2 minutes. Generally, 1 minute is enough for Clark College, meaning we make the safranin strong enough, but 1 minute is enough. But if your safranin solution is a little weak, you can allow it to stay for 2 minutes. You then dump the safranin off, and then you do the water washing. I should mention that if ever you're talking about the gram stain, you do not need to state that at every step you do a water wash. So you have to, if you're giving me the gram stain, you don't have to say flood with crystal violet, water wash, etc. Flood with iodine, water wash. You just need to say someplace in there, after each step, you do a water wash. Any question about that? After you stain with safranin, the color of the gram-positive cells is purple, and the color of the gram-negative cells is pink. You then water wash, and then you dump off the water, let the slide dry, and you can view it directly under the microscope. Even with oil, there will not be enough water to foul up the oil. All right, any questions about any of that? Here's a question for you guys then. Note here I said safranin gets in all the cells and stains them. Why are the gram-positive cells looking purple if they have safranin in them? If they have safranin in them, shouldn't these cells be pink or purple pink or both purple and pink?
Is it because saffron is in the purple dye?
I'm not sure I understood that. Safranin is in the cells, if that's what you're asking. And the crystal violet is in the cells.
Is it because it's already dark?
Yes, that's exactly it. The purple is a more intense color, and our eyes, human eyes, see the purple. We do not see the pink in the cells because our human eye, I don't know if it ignores it or just doesn't see it. Okay. If you were to look at the cell under the electron microscope, you could see, let's say this is the cell, you could see purple in one region of the cell and pink in one region of the cell. Okay. But to our eye, the cell only looks purple. And it's just because purple is a more intense stain, that's all our eye sees. We don't see the pink, but it is there. Any question about that? And I won't ask you that on an exam or something because it's kind of a trick question, but it's just curious that our human eye doesn't see the pink. But if we were to blow up the cell with an electron microscope, we could see the pink color in the cell. Already talked about that. Oops, went 2 again. All right, so the gram-positive cells stain purple, the gram-negative cell stained pink. Why? What's the basis of this differential staining? The differences are due to the differences in the cell wall. The gram-positive cells have a thick peptidoglycan layer, and the crystal violet iodine complex gets stuck in the thick peptidoglycan, so it does not wash out. The gram-negative bacteria have a thin peptidoglycan layer in their cell wall, and it's thin enough that the crystal violet iodine complex does wash out in the decolorization step. You should know the gram-negative cells also have a lipid polysaccharide layer as part of the cell wall. We'll talk about that. Well, I guess now. The gram-positive cell looks like this. Yellow being the cell membrane, purple being the thick peptidoglycan of the cell wall. And if we blow that up further, we'll see that the peptidoglycan is many layers thick in the gram-positive cell. And this is only showing you 3 layers, but in reality, the peptidoglycan is layer upon layer, and in the gram-positive cells, it's hundreds of layers, maybe even thousands of layers of peptidoglycan. Any question about that? Oops, I didn't want to do that. That's what I wanted to do. The gram-negative cell, when we look at the cell here, it has the cell membrane, which is lipid rich. It has a peptidoglycan layer, which is the purple, and it's thinner than the gram-positive cells. And then it has a lipid-rich region outside of that. And if we look here, we can see that there's the cell membrane, lipid-rich because of the phospholipids. And then there's the peptidoglycan layer. It's only showing you 1 layer, but in reality, it's many layers. I don't know how many, but it's much more than 1, but it's less than gram-positive layers. So it's certainly not thousands of layers. It's probably not even hundreds of layers, but it's much more than 1. And I don't know how many, so I'm not going to give a guess, but it's more than 1. It's only shown as 1 layer here. And the point is, it's a thin layer of peptidoglycan, and so the crystal violet iodine complex can wash out. And those cells do have a lipid-rich region on the outer part of the cell wall. And we'll talk more about that next time.
When the crystal violet binds, does it only bind to like the outer layer of peptidoglycan or to all the layers?
It binds to the entire layer of the crystal violet. Maybe that's not apparent here, but peptidoglycan allows water and water-soluble molecules to come right through the peptidoglycan. Crystal violet, as well as iodine, is water-soluble, so it comes right through the peptidoglycan. Any question about that? All right. I think I'll stop here. We only have a few more slides. I think we'll stop here and I'll see you. Oh, no, I don't have to stop. We'll go on just for a few more slides. There's no lab today, but I will be there from 6:30 to 6:45 just to answer questions. Let's see, is there anything here? Come on. I got it too big. I've already talked about that. We talked about that. All right, so here we're looking at a picture of the gram stain, and we have both gram-negative cells, like gram-negative spirochete, a gram-negative rod, and there are gram-positive rods. There's gram-positive cocci. Now, when we look at the gram stain, you will note that there are regions of the slide which are hard to interpret. For example, this looks gram-negative here, but these cells right there look gram-positive. And that doesn't look like cells. Okay, so how do you read this slide? Well, you need to read it in the good regions of the slide where the cells are individual. There's no clumps of crystal violet, and skip reading the slide in the bad regions. This region is bad because right there is a crystal of crystal violet, and it puts out some crystal violet, and that's what's making these cells purple or blue. And this region here, which is away from this crystal, the cells look pink. So this is a gram-negative cell, not gram-positive. And these are only purple because these cells are so close to a crystal of crystal violet. Do you understand what I mean by a crystal of crystal violet? Meaning the dye, which is a liquid solution, can crystallize, and then the bottom of the beaker of crystal violet, there may be a clump of crystal violet, and that's what that is. And you don't want to read your cells in this region. All right, any questions? There's a few other problems with the gram stain. You get the best results when the gram stain is performed on young growing bacteria. Do not use the gram stain on cells that are over 2 days old. Some cells do not stain well with the gram stain. And that's because they have so much lipid in their cell wall that the lipid keeps the water soluble dyes, meaning the crystal violet and the safranin, from coming into the cell. And so some cells do not stain well with the gram stain. They usually do stain, their staining is just lighter. And so the results are not always as clear-cut as theory suggests. And sometimes when you're doing the gram stain, it feels like you're pulling out your hair. Any question about any of that? All right, I'm gonna stop right here, and then we'll continue with this next point, which is really major, next time. Are there any questions? All right, if there's no questions, I'll say goodbye. And if you have any questions on anything, I'll talk to you in the lab. Or not the lab, but at 6:30. I'll be only there until 6:45.